video, I'll be discussing two topics. First is oxidation of pyruvate and second is fate of pyruvate. But before that, let me tell you what are the channels and proteins used by pyruvate to cross the outer and inner mitochondrial membrane. Pyruvate crosses the outer mitochondrial membrane using the voltage-dependent anion channel, also known as mitochondrial porins, that allows it to enter the intermembrane space. Voltage-independent anion channel or VDAC is a gatekeeper for the entry and exit of mitochondrial metabolites, thereby controlling crosstalk between mitochondria and the rest of the cell. Pyruvate seems to pass easily through the outer membrane compared to the inner membrane because pyruvate is negatively charged and quite polar, which makes it unfavorable to fuse directly through any membrane. As I said earlier, the outer mitochondrial membrane contains porins, which allow small molecules like pyruvate to passively diffuse through, unlike the inner mitochondrial membrane, which locks such channel and depend on active transport. But recently, researchers discovered a mitochondrial pyruvate carrier that pyruvate used to cross the inner membrane. Mitochondrial pyruvate carrier or MPC is a two-subunit carrier complex and a gatekeeper that is responsible for the pyruvate transportation and oxidation in the mitochondria. Pyruvate is produced by glycolysis in the cytoplasm, but pyruvate oxidation takes place in the mitochondrial matrix. So before the chemical reaction can begin, pyruvate must enter the mitochondria, crossing its inner membrane and arriving at the matrix. In the matrix, pyruvate is modified in a series of steps. Step 1, a carboxyl group is snipped off of pyruvate and releases a molecule of carbon dioxide, leaving behind two carbon molecules. Step 2, the two carbon molecule from step 1 is oxidized and the electron loss in the oxidation are picked up by NAD+ to form NADH. Step 3. The oxidized 2-carbon molecule is attached to coenzyme A, an organic molecule derived from vitamin B5 to form acetyl-CoA. Acetyl-CoA is sometimes called a carrier molecule and its job here is to carry acetyl group to citric acid cycle. There are three key fates of pyruvate, a product of glycolysis, which are mainly converted into acetyl-CoA, a substrate of citric acid cycle, lactate produced by eukaryotes in the absence of oxygen, and lastly ethanol produced in yeast. Under the aerobic condition, the majority of pyruvate is metabolized in the mitochondria to acetyl-CoA and ultimately to CO2 and H2O, which makes the product of the citric acid cycle and electron transport chain. Under anaerobic conditions, such as occurs in a muscle cell during strenuous exercise or in erythrocytes which lack mitochondria, pyruvate is converted to lactate, the ionized form of lactic acid, by the enzyme lactate dehydrogenase. Under anaerobic conditions in microorganisms such as yeast, pyruvate can also be utilized to alcoholic fermentation to convert pyruvate to CO2 and ethanol using the enzyme pyruvate decarboxylase and alcohol dehydrogenase respectively. Thank you for watching. I hope you learned something from this video. This is Eliza, our video Piranha, signing off.